So here's the continuation, part 2. Obviously, I haven't watched part 1 yet. This won't mean much to you, but we'll just continue, and I'll assume you have. Another useful word for you, one of these specific types of cytokines, is called interleukin. And that can be released by macrophages, which cause this clonal expansion. Sometimes it's also known as proliferation. Proliferation meaning making more of them. Let me give my spellings. Proliferation. There we go. Interleukin can also be released from T and B cells. It's a kind of general term for lots of the chemicals that are used. But anyway, let's just recap what we've got. Complementary receptor on the T cell links onto our antigen presenting cell. We get clonal selection. The right one is chosen with the correct shape, if you like. We make more of them. And then the T killer cells release their own chemicals. In this case, it's often toxic chemicals. Sometimes they release a chemical which causes cells to basically self-destruct, something called apoptosis or apoptosis. But the end point is that they will kill infected cells off. Okay. So again, there's a bit of cell signaling going on here. They will release a chemical that will tell your own cells to basically self-destruct, which is a normal part of cell cycles. Actually, cells do this all the time, except in this case, the process is being speeded up. So that's T cells or T killer cells. T helper cells, slightly different route. They still go through the clonal expansion. The clonal selection and the clonal expansion. Hope this makes sense. Yeah, I'm sure it does. And then they release interleukins. So here's an example of not just the macrophages releasing it, but this is the T helper cells. And this activates B cells. Now again, B cells do a lot more than I suppose it's suggested in the book. So we're going to keep it fairly straightforward. So you can see what's happening with these B cells. We'll go with the idea here that the B cell can only be activated by the right T helper cell. Some of them actually can be activated in other ways, but we'll go with this. T helper cell release interleukins, which switches on these B cells that also would have a complementary site exactly the same shape as this. We get the clonal expansion again, except this time we're getting a clonal expansion of the correct B cells. Some of these are then, and here's a useful word, differentiated, meaning the cell becomes something else. It changes. Basically, you've got genes on involved here that will be switched on, and it becomes a plasma cell. And it's the plasma cell that produces antibodies. So technically the B cell isn't producing the antibodies, it has to differentiate first and become a plasma cell. Some of these B cells would also become B memory cells. Some of the T cells can also become memory cells, so I should have put those on as well. But that's our process. Now it looks quite complicated. We've got a lot of cells involved here. We had the macrophage, we had the T helper cell, we had the B cell, and then differentiated into the plasma cell. All of this is mediated, all this is controlled by releasing these chemicals. And if it seems a bit complicated, that's just how it evolved, and it's pretty effective at keeping us fairly healthy. So it works. You might as well say, why don't you just have a B cell up here? Because you just don't. It just doesn't work that way. Okay. So really just to try and get the language right on this, because I think that's where a lot of the problems are, and a lot of people will drop points. Remember the difference between phagocytes and lymphocytes. Remember this idea of antigen presentation. Remember that it is receptor sites, complementary receptors on the surface of the membrane. Not only in this case for antigens, you also have complementary receptors for all of these chemicals like the cytokines and the interleukins. And just finally to mention, these are briefly mentioned in your book, another type of chemical used in cell signaling is monokines, which are released by macrophages, which can attract neutrophils. So when you injure yourself, some macrophages might be there picking up bits of pathogens and whatever. They release monokines that will attract neutrophils there to help to get rid of those pathogens. And monokines can also stimulate B cells to differentiate. So the other place this is useful is if you're getting questions about basic cells and it's talking about cell signaling. These are all examples of chemicals that would be detected by various different types of cells if they had the correct receptor site on their surface. If you don't have the correct receptor site, nothing's going to happen. But cells are constantly sending messages in the forms of chemicals, which is also really what a hormone is. It's a chemical message that is either received on the outside of a cell or in some cases on the inside.